Welcome everybody to this new edition of the Plenary Colloquium of the Italian Society of Statistical Physics. After a lapse of time where, for various reasons, the colloquia were over, we start again with the colloquium by Professor Guido Boffetta from the University of Torino. Guido is known to all, I guess, all people in the audience because we know him personally, but this doesn't prevent me to recall to all of you that Guido is one very important and renowned, internationally renowned expert in the field of turbulence and its applications to many research fields, like the presence of polymers, active matter, and all phenomena related to these very important facet of physical science, when it combines also with special ingredients that produce very interesting phenomena, as those that Guido is going to present to us in this plenary colloquium. I'm very happy and I want to thank him for accepting to give this colloquium to for SIFS. So, sorry. Thank you, Guido, and now it's your time.
Okay, so do you hear me, Roberto?
Yeah.
See? Yes, okay. Thank you, Roberto. Thank you very much. Thank you for the invitation and for this kind of presentation. So today I will give a kind of introduction to the dynamics and statistics of microswimmers in turbulence, and I will report work done in collaboration with several people, but in particular I want to thank Matteo Borgnino, Massimo Cencini, and Filippo Di Lillo for the long collaboration on these topics. So one slide of, let me see if it doesn't work. Wonderful. Yes. So one slide of motivation. So marine microorganisms, they live in a turbulent environment. This environment mediates all the crucial processes, including nutrient uptakes, we'll discuss a little bit, reproduction and predation. So one general question one can ask is how they do adapt to this turbulent environment, but maybe a simpler question, what are the effects of turbulence on microbial processes? And in particular, we discuss today how patchiness, so in homogeneous distribution and collective motion is produced by the combination of turbulence and motility. So one important example of the effect of turbulence in the microbial process is the case of Chemotaxis. I will not discuss Chemotaxis today in turbulence, but we know that marine bacteria, they have to perform a very difficult task because they have to swim up the gradients before they disappear due to the turbulent motion. And so typically they swim much faster, 10 times faster, roughly speaking, than enteric bacteria. And they have also different swimming pattern. It's mostly run and reverse instead of run and tumble. So this is selected to achieve this task. And in spite of the interest of this problem, there are relatively few studies of Chemotaxis in turbulence. This is just an example of a paper of by 10 years ago by Taylor and Stoker. They use a very simple model for Chemotaxis. And the idea at the end is that they found there is an optimal swimming speed, which is optimized the benefit of the increased uptake due to the chemotaxis and the swimming cost, because swimming at low Reynolds number is very expensive. So about swimming, let me remind you that in spite of the name, which means drifter, many planktonic organisms are able to swim and they do pretty well. And typically with some taxis, so chemical or light, so phototaxis or gyrotaxis, which I will discuss in details. So here there are two examples, but what are the advantages of motility in a turbulent world in which there is a velocity field which is typically much larger, the velocity is larger than the typical swimming velocity. So on the left, there is an example taken from zooplankton. This is a copepod. The copepod is this animal, is 1 millimeter, is a very sophisticated animal. He has 2 antennae. Here, by the antenna, it can detect emotion in the water. So the copepod are prey of several fishes. So here there is a juvenile cephalus. We want to feed on copepod. So this is the movie. And as the fish approach the copepod, you see this now in slow motion. The copepod is able to do this very long jump in which he reaches a velocity which is about 500 body length per second and acceleration of order 20 g. So relatively speaking, this is probably one of the fastest animals in the world. And on the right, there is another example, which is something which I will discuss more in detail. These are phytoplankton cell. So this is unicellular organism, 10 micron. It's much more simple organism. But also in this case, this is a real-time movie. So they swim very well. They swim fast, apparently in an erratic way. So why they want to swim? This is not clear, as in the case of copepods. So I have one slide of motivation why one is interested in phytoplankton, because phytoplankton are mostly unicellular organisms and they're responsible for the primary production in the ocean, and so they produce more or less 1/2 of the oxygen on Earth. So they are really very important. And to do photosynthesis, of course, they need light and nutrients. So in this movie is a satellite data which measure essentially the concentration of chlorophyll, which is a proxy for the phytoplankton concentration. And in 14 years, okay, you can recognize the seasonality, but what is clear from this movie is that phytoplankton is mostly concentrated at high latitudes, okay, while there is almost absent in the tropical ocean. And this is due to the fact, not by light, of course, but it's due to the fact that nutrients are more concentrated in the high latitude, okay, because of the upwelling for the deep ocean. There is also an upwelling close to the continental ships, so you see also a concentration close to the continent. But so the limiting factor here for the growth of phytoplankton is really nutrient, and nutrient is not only the limiting factor for the population, but also at the level of a single cell. This can be understood by this simple argument, which is taken by the nice book by Thomas Kjorbe. So since the phytoplankton cell is very small, the nutrients is transported to the cell surface by diffusion. So if you assume to have a spherical cell, the uptake of the nutrients, in particular as nitrogen, which is the limiting element in the ocean, the diffusive uptake is given by this expression, which is proportional to the background concentration of the nutrients, to the diffusivity, and to the radius, to the size of the cell, which A is the radius of the cell. Okay, and on the other hand, the metabolic consumption is proportional to a power of 2 or larger than 2 of the size of the cell. So the metabolic consumption grows faster, of course, than the uptake, and so there is a maximum size that the cell can reach, which if you put the number for the ocean, you get something which is of order of 50 microns, which is more or less reasonable. Okay, but there are larger phytoplankton cells. So how they can overcome this limit? Okay, one trivial way to overcome the limit is to duplicate. If you instead of one big cell, you do 2 small cells, you duplicate, you can start growing again. But there are other ways to escape this limit, and this is by motility, and also in the presence of a flow. Because in the case of motility, if you consider for simplicity a spherical cell in uniform motion, which can be due to swimming or sinking, there are general very classical results by Taylor and Batchelor that there is an increase of the diffusive uptake because the motility deformed the concentration surface around the cell, which are not spherical anymore. So this is quantified typically by what they call the Sherwood number, which is the actual uptake of nutrients over the diffusive uptake, which is this expression. And as a function of the Peclet number, which is a dimensionless number, dimensionless velocity. There are asymptotic results for small and large Peclet number, but people in the oceanography usually use this empirical formula which interpolates between the two. Okay, this is empirical. There is not exact result in general case. And you see that when the Peclet number, so velocity, or swimming, for example, swimming velocity is sufficiently large, the Sherwood number can be 2 or even 4, which is not a huge number, but from ecological point of view is very important. You have the double of the uptake. And also the presence of the flow, in particular turbulence, can deform this concentration surface around the cell. And also in this case, we have an enhancement of the uptake, which is again given by the Sherwood number. But now the Peclet number is defined in a different way because there is not a mean velocity, it's defined in terms of velocity gradients. Okay, and epsilon represents the turbulent intensity, which in the ocean range between 10 to the minus 8, 10 to the minus 4 square meter per second cube. So, okay, look at this plot. This is the Sherwood number, so the increase of the uptake induced to turbulence as a function of the cell radius and as a function of the intensity of the turbulence in the ocean. And you see that there is a significant enhancement of factor 2 or 3 only for large cell and for strong intensity in turbulence, which correspond to storm or breaking waves. But I want to remark that this result is just a kind of mean field result because all the turbulence is parameterized by a single number, and we know that epsilon has very large fluctuation in real turbulence. So locally, you can have a Sherwood number which is much larger than this turbulence. So another slide about the scale that we consider for microorganisms in the ocean. So we consider phytoplankton. These are unicellular, as I said already, between about 1 micron and 100 micron. And in all cases, they are well below the Kolmogorov scale, which is the smallest active scale in the turbulent ocean, which is about order of millimeter, few millimeters typically. And also they are smaller than the Bachelor scale, which is the smallest scale at which the nutrients, so the spacescalar, are mixed by turbulence. So from this point of view, we can consider marine microorganisms, phytoplankton, as a point particle which are transported by the flow. Now, if the particle is simply transported by the flow, we can use what is called the Lagrangian description. So the velocity of the particle, the position x represents the position of the particle, the time derivative of the position is given by the fluid velocity in the position computed at the particle position. So it's an overdamped system, so there is no inertia. So formally, this is a three-dimensional dynamical system which has a contraction rate in the phase space which is 0 because the flow water is incompressible. So the divergence of u is equal to 0, and so we expect that because it's incompressible, that the particle, the trajectory, will visit all the phase space in the, which is a configuration space, in a uniform way. So we expect that the blob of phytoplankton will distribute, as shown in this movie, uniform in the space. But of course, this is not true if the flow is not incompressible, the system becomes dissipative, and so we expect in that case that the trajectory concentrate on a subset, which is typically a Lagrangian subset, a fractal subset. So an important point is that I want to make is that it's possible to have a dissipative dynamics in incompressible fluid where the particles are not simply transported by the flow, because, for example, the presence of an intrinsic motion like swimming or an external force like gravity. Very often, this model can be described by a simple particle transported by an effective velocity field, which is given by the fluid velocity plus an additional velocity which represents the intrinsic motion. And the velocity field is incompressible, but this is not the case for V, which depends on the particular problem, but typically the divergence is different than 0, so we have dissipative dynamics, and so we expect that the particle concentrate on a fractal cluster. So this plot shows an example of, which is not for swimmers, this is for inertial particle, which can also be written in this way, in some particular limit. And you see there are very a nice clustering, which somehow is reminiscent to the patterns observed, for example, in flocking or in schooling. But I want to make clear that the mechanism is completely different because there is no particle-particle interaction here. And so the collective behavior, if you want to call it in this way, the collective behavior is produced by the presence of a flow that transports the particle. So this was more or less the introduction. Let me move now to the specific problem that I want to discuss, which is how turbulence induced clustering in gyrostatic organisms. So first of all, what is gyrotaxis? So gyrotaxis is essentially the ability to swim along gravity. Many microorganisms, phytoplankton cells, are able to detect gravity. And I give you here the example of Chlamydomonas, which is a well-known organism. This is a unicellular flagellate with more or less 10 microns in size. He has 2 flagella. He swims very well. It was also the movie that I showed before was Chlamydomonas. This is slow motion, about 10 times smaller than the reality. But they are quite sophisticated. They also have an eye spot, so they can detect light and make phototaxis. But what is important here is that the chloroplast distribution inside the cell is not uniform, so they are bottom heavy. And so therefore, the center, look at this scheme, the center of mass is displaced with respect to the center of buoyancy. They are neutrally buoyant. And so there are 2 forces which produce a torque, and this rotates the cell in the vertical direction. So they naturally swim against gravity. They naturally swim upwards. And people from ecology, they tell you that they want to swim up because they want to reach the surface of the sea and find light, essentially. And then during night, they stop swimming, and because they are a little bit heavier than water, they sink in the deep water where there are less predators. But I want to convince you in the following that a combination of this swimming, the gyrotaxis, together with the flow can produce a non-trivial motion and non-trivial concentration, so much more complicated than going up and down in the vertical in the vertical water. Okay, so the model for gyrotaxis was produced by Kessler many years ago. It's very simple. So the cell is very small, so it's a point particle, as I discussed before, which is transported by the velocity plus an additional swimming velocity. Vs is the swimming speed, which in this case is 100 microns per second, and the swimming direction is determined by the second equation, which has 2 terms. The last term is the rotation of the cell with the local vorticity, so the local how the water rotates essentially, and this is the gyrotactic term which orients the cell in the vertical direction. V0 is the characteristic velocity for this process, and the relaxation time for the orientation is given by V0 over G, which is of order of few seconds in typical cell. Okay, so the model is very, very simple, but can explain, for example, a remarkable feature of this organism, which is gyrostatic focusing. So this is from the original paper by Kessler. So if you put a suspension of gyrostatic organism in a pipe flow, this is a downwelling pipe flow, so a laminar flow. So you have a parabolic profile for the velocity field. The combination of gyrotaxis and vorticity, because you have a velocity gradient, makes the cell to swim towards the center of the cylinder, towards the axis of the cylinder. So you see a concentration or a focusing of the swimmer in the center of the cylinder. You see here in this picture is the black line. On the contrary, in upwelling flow, the combination of the two makes the cell to swim towards the boundary of the stereo of the cylinder, so they accumulate on the boundary. And this is the gyrostatic focusing. So recently, together with Massimo and Filippo, we propose a generalization, a refinement model of gyrotaxis, which takes into account. So the idea was that in turbulence, you have not only the gravity, but you also have acceleration, which is due to the fluid motion. And in turbulence, acceleration can be of order g or even larger. So in this case, we want to take into account all the possible acceleration in the fluid, so we simply generalize the Kessler model with this additional term. But in this way, we have a new prediction. For example, a very simple prediction in the case of solid body rotation, which is, if you want, is a toy model for a vortex. So if you put a gyrostatic particle in a solid body rotation, because you have a centripetal acceleration, if A is sufficiently large, you have that they they orient to swim toward the axis of the cylinder, so we accumulate around the axis, but not for the gyrostatic focusing, but simply because of centripetal acceleration. So we perform some laboratory experiments just to check that this model works with a real swimming cell. So we put a suspension of Chlamydomonas in a rotating vessel. We illuminate with a laser sheet, and so I show you some pictures taken with a normal camera, what you see here. So this is a two-dimensional section given by the laser sheet of the experiment. At the beginning, they are uniform distributed. So the dashed point, the white point here, the white point here represents the axis of rotation of the cylinder. And as the time goes on, they swim toward the axis, so they concentrate in the central part of the vessel. And this is due to the fact they are swimming, not to other physical effect, because if you repeat the experiment with non-swimming cell, you don't see any concentration. So we can make some quantitative prediction from the generalized gyrostatic model, but okay, I don't want to enter too much into detail, but you have to consider the fact that there are, for realistic organisms, there are some stochasticity, which is essentially a biological stochasticity due to the fact that they not always swim in the same direction. Sometimes they turn on the left, on the right. So we rewrite the model in a stochastic version, in which there is some angular noise here, and dr is the angular diffusivity, and then you write the Fokker-Frank equation, and then assuming fast relaxation of the swimming direction to the local equilibrium distribution, you end with an equation for the density with an explicit expression for both the drift term and the diffusivity tensor. And in some limit, which is essentially for small r, you have a prediction for the equilibrium distribution of the radial distribution of the cell, which is simply at the end is a very simple a Gaussian with all the parameter which is determined by the physical and the biological parameter. And so we made from experiment we made some quantitative measurement using fluorescence imaging of this. This is exactly the experiment as before, but now we use the fluorescence induced by chlorophyll, and so we can have a quantitative measurement of the density. This picture is very nice. This is time zero. Each dot that you see here is one single cell, so you can average over several realizations, and then you take the radial distribution of the concentration. This is at initial time, this is at final time, and if you plot the distribution of the cell as the times as a function of time, at the beginning they are uniform concentrated, which are the red square here, and then at the final time you reach a stationary distribution, which is given by the blue triangles. And the line represents the prediction by the stochastic model, which I want to stress this is without free parameters, in the sense that the parameter for the swimmer, which are the swimming speed, the orientation time, and the angular diffusivity, are taken by the literature. So we have no free parameter here, and they fit very well the data. So now we want to, this is just a toy model for a vortex, so we want to understand how the gyrostatic swimmer behaves in turbulence. To do that, we use direct numerical simulation. So we couple the gyrostatic model with a velocity field which is produced by a numerical integration of the Navier-Stokes equation in a cubic box, which you see here in this movie. We use periodic boundary condition in order to avoid any problem induced by boundaries. And the point represents the position of some of the swimming cell. Sorry. Okay. Which at the beginning at time zero, okay, we restart at time zero, they are uniform distributed, but you see that they concentrate in some particular region of the space that evolve in time because there is an underlying turbulent flow. So indeed, formally, this is a six-dimensional dynamical system, which is dissipative. Okay, if you compute the phase space contraction rate, it's proportional to this quantity. And so it means that as P orient in the direction of the local acceleration, this quantity becomes negative, and the swimming cell are expected to concentrate on a fractal subset of the phase space. Okay, so we want to characterize this concentration. So these are some example of the distribution, the stationary distribution of the cell for different value of the swimming parameter. I forgot to mention that there are two important dimensionless parameters in the model. One is the swimming number, which is the swimming speed normalized with a Kolmogorov, so a typical velocity in the flow. And the other parameter is the stability number, which is the ratio between the orientation velocity and another typical velocity in the flow. So these are three examples. The first one is for non-swimming cell. If they do not swim, they remain homogeneous distributed, and these are for other two different value of the parameter. So you have different degrees of clustering. So we want to understand how the clustering depends on the parameter, and if possible also where in statistical sense the cell cluster. So to characterize clustering, we use because we expect the clusterize in a fractal set, we compute the fractal dimension, in particular the correlation dimension of this snapshot for different value of the parameter, and they are plotted in this plot as a function of the stability number for different swimming speed. Okay, so you see that in general, increasing the swimming speed, when the correlation dimension is equal to 3, it means homogeneous distributed, so no clustering, and less than 3 means clustering. In particular, where D is equal to, it means that they concentrate over a manifold, which is essentially equivalent to a surface. Okay, so the clustering increase with the swimming speed, but you see here that there is an optimal value of the stability number of order 1 to have the maximum clustering. And indeed, it can be easily explained that both in the limit of very small stability number, very large stability number, we cannot expect to have any clustering. Okay, so this is the two limits are so under control, but we don't know. In principle, we didn't know where is the maximum clustering. So we can try to understand how clustering depends on the stability number. So I rewrite here, this is the gyrotractive model written in dimensionless form. So this is the swimming number, the stability number. And we can make an expansion of the second equation at the first order in psi. Okay, so essentially you require to have equilibrium alignment. So this term is equal to 0, and then you solve this equation for at the first order in psi. So you obtain an exact expression, an approximate expression, but an explicit expression for P, and you use in the first equation. And now you have reduced your system to a three-dimensional system, in which you have an explicit velocity, which is an effective velocity, which is not divergence. So there is a contraction in the phase space, and the divergence of the effective velocity is proportional to 5 psi Laplacian of the vertical velocity. So from this result, we can use a very general result of clustering the weakly compressible velocity field, which is summarized here. If you have a compressible field, a weakly compressible field, so delta is a small parameter, you expect that the tracer will clusterize in a fractal set with a codimension proportional to delta square. So it means at the end that we expect that the fractal dimension is equal for small psi is equal to 3 minus a constant which we cannot determine with this argument, 5 psi square. And so this suggests that instead of plotting as a function of psi alone, we should try to plot as a function of the combination phi times psi. And indeed, if you replot the data in this way, you have a perfect collapse on the left part of the plot, and also the quadratic profile fit pretty well the data. So we can also understand where the cell cluster is in a statistical sense, because the divergence is given by essentially minus the Laplacian of the vertical velocity. So we expect that they concentrate a region where the Laplacian of u zeta is positive. But now, if you remember that this is a technical point of homogeneous isotropic turbulence, you have that the energy dissipation rate, which can be written as the correlation between the vertical velocity and the Laplacian of the vertical velocity, must be positive. It means that the region where the Laplacian of the vertical velocity is positive corresponds to a region of negative vertical velocity. So at the end, the prediction of the simple argument is that gyrotractive swimmer accumulate in a region where the vertical velocity is locally negative. negative, so downwelling region. If you want, this is a generalization of the result of Kessler. This was for a laminar flow. They concentrate in downwelling flow. In a turbulent flow, it's much more complicated, but we expect that the particle concentrate in a region where the vertical velocity is negative. And so this is the last movie that I want to show you. It is a vertical section of the velocity field. Negative velocity is yellow. The point represents the position of the swimmers. At the beginning, they are uniformly distributed, and you see that they mostly concentrate in yellow region. Of course, everything is dynamic, then the yellow region disappears, so they have to find another downwelling region. But in general, if you compute the average of the velocity, the vertical velocity computed over the particle position, which is not uniform, you have a well-defined negative average value, while the Eulerian value, the global value in the volume is by definition is 0. So at the end, we reach a kind of paradox, no? Because we have a swimmer that want to swim upwards, but they concentrate in the region where the water goes down, downwelling region. So is this a paradox? We don't have an explanation for this, but I want to remark that this maybe is not a paradox because the behavior is not generic, because it also depends on the shape of the cell. So everything is quite complicated. So so far I consider only spherical cell, but we can extend the model to non-spherical cell, for example, to ellipsoid. And so in this case, we have an additional term in the orientation. This additional term is due to the fact that if the cell is not, the particle is not spherical, there are some force given by the rate of strength tensor S, which is nothing but the symmetric part of the gradient of the velocity, which orient the cell. Alpha is a parameterization for the shape, so alpha equals 0 is a spherical cell, and so this term disappears. Negative alpha is a disk-like particle, and what is more interesting for application is alpha larger than 0, which are elongated particle. Alpha equal 1 is a rod, is a one-dimensional rod. Okay, so for this model, there are some analytical prediction by the group of Mehling and collaborator. They do not use real turbulence. They use a stochastic velocity field which has some properties similar to turbulence. They do perturbative analysis in the Kubo number. So the story is quite long also in this case, but they show that rod-like swimmers, so with alpha larger than 0, they can accumulate either in upwelling or downwelling region depending on their velocity. So the result is shown in this plot. The pink line, the pink point, represents the negative vertical velocity over particle, over elongated particle as a function of the swimming speed, and they change sign when the swimming speed is sufficiently large. So we check if this behavior, which is quite complicated, can be also observed with real turbulent flow. So we perform numerical simulation of this extended model together with this term, which is called, by the way, is called the Jeffrey term. It's exactly 1 century ago. And so these are the results. So this the average velocity over the swimmer. Now the swimmer are rods. Okay, the swim in turbulent flow as a function of the swimming speed. So if they are if the swimming speed is not too large, they accumulate in the downwelling region, so the average velocity is negative. But if the swimming speed is sufficiently large, the average velocity change sign, so it means that they accumulate in the upwelling region. So there is a downwelling transition according to the shape of the particle and to the velocity of swimming. So the situation is quite complex in this case, and we have also some. Okay, the agreement is not only qualitative with an analytical prediction. In the limit of small swimming number, we also have a quantitative agreement with the analytical prediction. So we replot our data here, compensated with the prediction, and so the perturbative analysis predict that they stay on this dashed line, and for small psi, the agreement is very good. And so I was very fast because this is the end of my talk, and I hope that I have stimulated your interest essentially in the fascinating world of microorganisms swimming in turbulence. And if you want to know more details, you can have a look at this list of paper. So thank you very much again, and thank you for your attention.
Thank you, Guido, very much for your nice talk. I hope that someone has any questions for Guido.
I can ask a question, maybe, or a couple to Guido. Can you hear me?
Yes.
I was wondering, two questions, applied questions about real data. In the real data, I mean in the field. Do we have any hint that this preferential accumulation on the downwelling regions is indeed happening also in the real ocean, or?
As far as I know, no. In the field, no, for sure. And I don't even know if there is any indication in laboratory experiment. I don't think there are laboratory experiments with a kind of turbulent-like flow in which they do observe this accumulation. There are some experiments. Also, the Stoker group, they did some experiment, but with a very simple flow, which is not turbulent. Okay, which is essentially are two rotating disks that produce some upwelling, downwelling flow, but it's a very simple flow.
The other question, I remember typically when you see these pictures from satellites, they typically tend to show also different animals, different populations of different animals that are. Do they compete? I mean, is there any indication that indeed? This
is a very nice question, which I'm not an expert, of course, of that. But what I read is that, this is one of the paradox of plankton, how they call, in the sense that plankton, there are very, very different species, and they essentially need the same. They need light, and they need the same nutrient to grow. And so because they are mixed, the traditional idea is that turbulence mix everything together, and so they should compete. So why there are several species? Because one species should dominate over the others. So the fact that they have accumulation in different regions, which depends on the specific parameter of the cell, is one possible way to escape this paradox. No, one possible explanation, because if they concentrate in different regions, even very close region, but they do not compete directly for the same.
So is the flow itself that helps them to stay separate?
Exactly. This is the idea. Which is very nice, because means that indeed I put at some point is there is no life.
Do they need to stay together because of population? I mean, is there an advantage to grow together in the same region? Or?
Some of them do sexual reproduction, so in that case they need to stay together, but they can also do not sexual reproduction, so in that case no. And then depends on the species. Thank you.
So I wanted to ask a similar question. So if there is a reason which is ecological for this behavior, so your answer is that is this a possibility to be to compete in different part of the so is this the reason? I mean, the selection reason for this behavior?
Yes, so one possibility is that since if you swim, you are not uniform distributed, it means that if you swim with a little different parameter, you reach different local regions, local region in the ocean, so you don't directly compete with other species. But this is a very general statement. I mean, in particular for the case of Chemotaxis swimmer, why they go in the welly region, I really don't understand. I don't think no one understands really why. It is not generic because they are elongated, it behaves differently, but in any case, maybe it's just a coincidence, which is not important. I mean, but I don't know.
Is the disadvantage to be spherical?
Is there?
No, I don't know. I mean, that's
this I don't know. They not always swim, by the way. Part of the day they don't swim, so they a little bit sink because a little bit heavier than water typically. So they quite complicated.
But I mean, just a question, but are there probably it's just a matter of collecting data about the behavior of these different species to understand how they do interact with each other, because I'm completely ignore if which is the state of the art in the collection of data about the behavior of these species. Because in order to make them all live together, there should be really a very interesting aspect to be explored to understand if there are different equilibria that in some sense can coexist in these different species according to the way they move. But is it just a matter of satellite observation, or there are...
No, okay. So no, the point is that the scale is very, very small. Okay, our simulation are a few liters of water, if you want to take it. So you need to have local experiments. As far as I know, until a few years ago, the data for phytoplankton were taken by you go in the water with a ship. You use a bottle and you take up from the bottle. So you average over a column of several meters. So of course you cannot use this kind of data to detect possible this behavior. I think that probably laboratory experiments are more important. No? In laboratory, you can have under control everything, and you can have a precise measurement of the distribution of different species before going to in the ocean. And there are indeed people that are doing laboratory experiments, but I am not aware of an experiment with a realistic turbulent flow yet.
But as I understand, according to the comment by Luca and Raffaella, it seems that the turbulent flow plays a sort of selection role in order to make compatible species live together, and all the other cannot do it. So in some sense, a selection already appeared, but once you have conquered your space, you keep it. And
but all the species are affected by turbulence. Also, for example, you see in this is a demological picture. These are diatoms. No, diatoms are about 1/2 of the phytoplankton. They do not swim. They cannot swim. But for example, most of them they can regulate. They are heavier than water, and they can regulate buoyancy. So they can go up and down. And also because they are heavier than water, they do not simply follow the streamline of the ocean. So also diatom can concentrate in particular region. So I think that most of the species can in some sense escape the streamline and find a kind of niche in which they can grow without competing with the other species.
Are there other questions? Apparently there are no more questions. So
okay,
I would like to thank Guido again for this very nice seminar, and thank you.